Mine results obtained, project publication and future collaborations. We look at the depth of coverage across the map fragments, identifying several highly sequenced chromosome regions, e.g. the RNA multi-copy regions, that will probably be analyzed separately. Coverage per individual is useful for our purpose and also for SNP calling. Though, despite experimental attempts to maintain equal temperature amounts per individual, we still detected up to tenfold variation in the map read count, probably simply due to the currently unavoidable stochastic factor and measurements inaccuracies. Another important experimental finding was as follows. Although the RL library preparation include a size selection step that target 300 until 500 BP fragment only, a high proportion of fragments obtained were smaller than 300 BP. We suggest this is due to smaller fragments forming sequencing cluster on the Illumina chip more readily. This knowledge is useful for ongoing library preparation is in Von Willis Group, Evolutionary Botany Lab, University de Natal, as it will affect the overall coverage prediction and so affect the optimum number of individuals put in a single library. By examining the fragment count output together with the reference map fragment display, we found several issues that required troubleshooting at the data processing level. We discovered the adapter offering installed fragment were inverting the fragment count. Since read with different endpoints were counted as different fragments, near the end of the week, we also discovered some discrepancies that seemed to be due to incorrect borrowed convolution. To verify this and to perform better adapter timing, we are currently reprocessing the entire raw data set with an alternative barcode convolution, renaming method, and including adapter timing and quality control. To proceed with our aim of looking at methylation difference, we select all fragments with no or very few reads with one enzyme, but many reads in the other for the same individual by looking at these fragments and their close neighbors in present. We could distinguish probable methylation at some sites. One important initial observation is the heterozygosity in methylation state seems quite common. This can be detected when a long HPLL fragment with an initial internal CCGG site in found, implying the internal CCG site in methylate, together with internal fragments implying the internal CCGG in unimethylate, whereas the long fragment is not produced using MESPL. Methylation sensitive A methylation sensitive AFL methylation sensitive AFLP M as AFLP studies would interpret this pattern when present with HPL discussion but absence with MSPL discussion as evidence of a methylate internal C in the regression site. A.G. Solomon et al. 2008, Richard et al. 2012, which would obviously be incorrect in many cases according to our result. We see this as an important and highly publishable result that will be the main focus of our first publication on the topic. We also discuss and plan the next steps for this analysis, namely, first, including in the Python script a check in the read, where possible, the presence of internal regression site found in the reference sequence. Second, rerunning the script with the newly processed data for optimum accuracy. Third, deciding how to rate fragments under 300 BP or whether the coverage will be set for the small size range. For quantifying a threshold of certainty for assigning rate patient's absence, taking into account the mean and variance in overall average for the individual enzyme or combination. In summary, this visit was an extremely valuable contribution to our ongoing collaboration. We planned and carried of initial step in the analysis of next generation sequencing data. Once the workflow reruns has finished, we will be able to progress further in quantifying the utility of this method to conservation genetic studies in comparison to current alternatives like methylation sensitive AFLP. We expect we expect to have a manuscript ready to submission on the topic in late 2013. So our collaboration will continue at least until this point. In addition, I'm currently applying for further funding with the aim of applying this sequencing analysis method to experiment I have performed that investigate heritability to DNA methylation and its inducibility in plants under drought stress.